So today's topic will be cell cycle and cell division. Now in this slide we will be knowing what are the learning outcomes. So at the end of this lesson, what we will be knowing. So we should know what is cell division, how do cells divide or why do cells divide, and what are the types of cell division, what is mitosis and its stages, what is karyokinesis, what is cytokinesis, what is a cell cycle, and what is the importance of mitosis. So here is a huge lot of things. Please don't get panicked seeing so many things we need to learn in this lesson, but all, if you learn it systematically, it is quite easy, and I'll make it sure that it should be in a very lucid and easy manner and you get to understand it. Now let us first concentrate on what is cell division. Now our next slide will be on cell division. Now in this slide we are concentrating on what is cell division. Now as per cell theory, we know that all cells are derived from the pre-existing cells, and from the basic like third and fourth standard we have studied that all living beings are made up of cells, and cell is the structural and functional unit of life. Now if you concentrate in this diagram, it's a diagram which is showing a cell which is having a nucleus and is after different stages is being divided into two different daughter cells. So this is how generally a cell divides. So it is formed from a pre-existing cell. So a pre-existing cell, follow my pointer, red pointer here, a pre-existing cell is here, from which after different stages which we are going to learn in this video, through different stages we come to this point where it is divided into two different daughter cells, and which are almost similar to the parent cell or the previous one. Now what is then cell division? It is a process by which the cells produce new cells. Now cell division, absolutely it is different in prokaryotes as well as in eukaryotes. Now you know, I think, what are prokaryotes and eukaryotes. Prokaryotes are mainly the unicellular organisms. Like initially when life started, the unicellular organisms were bacteria and all. So cell division in them is different from those in the eukaryotes like fungi, protists, plants and animals. Now let us move on to the next slide. Now here we will concentrate to know that why do cells actually divide. So what is the necessity of cell division? Why is it required? So a cell should grow. For the growth of a cell, it should divide. If there is a damage in a cell or a damage in a tissue, for the repair and replacement of the damaged cell parts, cell division is required. It is also required for the repair of tissues. It is also required for reproduction of species. So from a single cell to a many huge number of cells, if we want to multiply the number of cells for several different reasons, we need to divide the cell. So for that only cell division is required. So let us see what we are having in the next slide. Now, what are the types of cell division? Now, generally we know there are two types of cell division, mitosis and meiosis. I think in standard 7 you have studied a bit about very basic idea of cell division you are having, though in 10th you will study it in a huge detailed manner. So we know gradually that we are having two types of somatic cells and reproductive cells in our body. So somatic cells, they form the part of the organism, the body cells. So there the somatic cells, when they are required for the growth and development of somatic cells, the cells divide through mitosis, and the reproductive cells or the sex cells which produce gametes, there the type of cell division is called meiosis. So is it clear for everyone till now? Whatever I have discussed from the first slide, I think it should be clear. It's a very basic idea of cell division. So mitosis is generally occurring in the cells which are somatic cells or the body cells, and meiosis is occurring in the cells which is reproductive cells or sex cells. So as of now, I think it is clear. If you are not getting it clear, so please write in my comment box any queries or any doubts so that I can fulfill in the next video when I am posting. I'll clear your doubts and I'll fulfill all your queries in the next video. So please continue with the lessons. Let us see what is there in the next slide. Now this is mitosis. Now what is it? Mitosis is actually the equational division where one parent cell divides to form two daughter cells. Now you see my pointer here. Here is one parent cell which is dividing into two daughter cells, and if you see these daughter cells, they are identical to the parent cells, like they look exactly to the parent cell, and the same number of chromosome the parent cell is having is maintained at each stage of this mitotic cell division. Now here you can see this two cells are formed from this, and they are having identical characteristic features, and at each stage they'll have the same number of chromosomes. Now mitosis is having different stages. Now starting from interphase, the other stages are prophase, metaphase, anaphase and telophase. So this is prophase, then prometaphase, then there is metaphase, then anaphase, then telophase. Then this is cytokinesis. We will learn that also when the cytoplasm divides. And first 4 stages are under the stage of karyokinesis. So this is called the equational division, mitosis, where the one parent is dividing to form two daughter cells. So I think this is clear with all my students, and I can move on to the next slide. So here just a quick view about the phases of mitosis. So in the previous slide I have told you already there are different phases of mitosis. The first one is the interphase, then comes the prophase, then there in the previous slide, if you remember, there was a picture which was showing prometaphase. Now here we have not written that as a prominent phase, as the main 5 phases are present in mitosis. So interphase, prophase, metaphase, anaphase, and the last part is telophase. Now this all parts we are going to study in details in this video. So let us start with the first phase, interphase, and what are the things happening in that interphase we will know. Now here is the first phase of mitosis, or the interphase. Now here in this diagram you can see what is happening. Now just look in this diagram, follow my red pointer. Now this is the phase, a growth period between two successive cell divisions of a cell. Now we can say that it is a preparatory phase of the cell, when the cell is preparing to start the cell division, or when the cell starts dividing. Just before that we are entering the interphase. So after studying all these phases, I'll tell you what is a cell cycle and how these phases are under this cell cycle also. Now first let us concentrate here. So this is a resting phase where no external change in chromosome is visible. Now what is happening? The cell is metabolically most active and preparing itself for synthesizing the DNA and RNA to almost double the amount. So it is preparing for the cell division, it is metabolically active, and it is preparing for the RNA and DNA synthesis to double its amount. The volume of the cytoplasm increases, the volume of the nucleus increases, and the chromosomes are not yet distinguished. Now it is clear, this is the first preparatory phase, or the resting phase, where we can't see any external visible changes in the chromosome. So this is the phase where the volume of the cytoplasm or the nucleus increase, and the cell prepares itself for DNA and RNA synthesis. So I think it is clear for everyone. So let us move to the next part. Now let us start with the prophase. Now this is the first longest phase of mitosis. Now what is happening here? In this phase, the chromatin materials undergoes condensation. So what is happening? What does this term condensation means? They become short and thick, and the changes into thread-like structures called chromosomes. Now here we can see thread-like structures called chromosomes are formed, and they are much thickened. Each chromosomes here duplicates to form sister chromatids. Now chromosomes with two sister chromatids, you can see here, they are thickened materials, and each chromosome duplicates into two sister chromatids, and this two sister chromatids lie close to each other, and they are attached in a central point which is called the centromere. So here the point is called the centromere. They are actually attached in this point, the two sister chromatids. Now as prophase advances, what we will see, the chromosomes become much more shorter and much more thicker, more condensed. Here you can see they are thicker and shorter, but as it gradually advances, we will see them more shorter and more thicker. And in animal cells generally, the centrosome initiates and regulates the cell division, and it splits into two small round bodies called the centrioles. The centrosome here is shifting to the opposite poles, and it splits into two small bodies called the centrioles, that we will see in the next stage. They are called centrioles generally, and the centrioles migrate to the opposite poles. These centrioles will be migrating to the opposite poles of the cell. So what is happening here? First, this materials, these are condensing. This chromatin undergoes condensation. They are becoming thick and short. They are divided into two sister chromatids, which are joined at a central point called the centromere. Now as the prophase advances, they become more shorter and more thicker, and the centrosome, it initiates the cell division. They form centrioles which move towards the opposite poles, and that we will see in the next phases of cell division. Now is it clear this phase? Please go through your textbook whenever I am teaching you. As soon as your video learning through the video is completed, just open your book once and read it. And any doubts, any queries, ask me in the comment box. And don't forget to subscribe to my channel, and please do like and share my videos. Now you will get different links on the top so that you can see my other videos too. Now let us see what is there in the next slide. Now in this slide we are continuing the prophase only. Now in previous slide we have told that the centrioles are formed. Now we didn't see the diagram of centrioles there. Now in picture I can show you the centrioles. These are the centrioles which are formed, and they migrate towards the opposite poles. You can see the previous picture was the centrosomes were here. Now from them the centrioles are formed here. You can see this green things are centrioles, and which are moving almost towards the opposite poles, and asters are formed around the centriole at each pole. You can see asters are formed, and between the two centrioles there are spindle fibers. Now these are spindle fibers. Now I think you are following my red pointer. So spindle forming has started already at the end of the prophase almost. So between these two centrioles, spindle is formed, spindle fibers are formed, and gradually the nuclear material and the nucleus disappears at the end of the prophase. So the nuclear envelope, we can see the nuclear envelope has started dissolving. So the nucleus will also dissolve and disappear, and the nuclear material will also disappear at the end of prophase. And now this duplicated chromosomes here, the yellow and red things, will start moving towards the equator. So the centrioles have also started moving towards the pole. There are spindle fibers formed in between the two centrioles, and the chromosomes have started moving towards the equator. So this is the full phase of prophase, and it is the longest phase where many changes in the cell occurs, which are actually visible. We have seen so many changes here, and please try to remember that, so that when we are moving to the next phase, we can specify that what were the changes in prophase, and now what are the changes we are going to see in the metaphase. Here comes the metaphase. Now after prophase, in this phase we can see all the chromosomes have arranged themselves on the equatorial plane region. So see here my pointer. Here all the chromosomes have arranged themselves in the equatorial region. And how their arms are facing? Their arms are facing towards the poles. Now each chromosome is, if you see my pointer, you can see that each chromosome is attached to a spindle, and all they are arranged in such a way that the arms are facing towards the pole, and that all the centromeres lie in this equatorial region. So that in next phase we will see that they are moving towards the pole. So this metaphase is quite easy phase. Actually it is a phase between the prophase and the anaphase. So it is the meta, which means in between, and metaphase means the stage in between the prophase and the anaphase. So it is clear to everyone, right? All the chromosomes are arranged in the middle region or the equatorial plane. The arms are facing towards the pole, and each chromosome is attached to a spindle fiber at its centromere. So this centromere point is attached to the spindle fiber. So let us move on to the next phase quickly, which is the anaphase. Now this anaphase is actually the shortest phase of mitosis. What happens there? The centromere of each of the chromosome divides into two halves. See, they have divided into two halves, and the sister chromatids are separating, and they are moving towards the opposite pole. The centromere has divided and the sister chromatids are moving towards the opposite poles. Now due to the contraction of spindle fibers and due to the repelling force developed between them, so each have started repelling the other sister chromatid, and they are moving towards the opposite pole. Now depending on the position of this centromere, we can see that these chromatids are having different shapes. Either they are looking like V, or they are looking like J, or they are looking like U. So these chromosomes always are appearing in different shape. And how the anaphase ends? As soon as these chromatids are reaching the opposite poles near the centrioles, so then this anaphase is completed. So the anaphase ends when all the chromatids reach the opposite poles. So it's clear to everyone, right? It is a very easy phase where the centromeres are dividing and the sister chromatids are moving towards the opposite pole, and they are having different shapes like U, V and J. So next comes the telophase, or the last phase of mitosis. Now here comes the last phase of mitosis, which is the telophase. Now telophase, what happens? It is actually the exact opposite of what is happening in the first phase of mitosis, or the reverse of prophase. All the events mostly are the reverse of prophase. The chromatids actually, they start uncoiling and they elongate and change into a network of chromatin threads. See, you have seen the chromatids or the daughter chromosomes, they start uncoiling and they elongate and they change into a network of chromatin threads. You can see the chromatin threads have started forming. Here the nuclear membrane again starts gradually reappearing. So in the prophase we have seen that the nuclear phase started disappearing because the nuclear and its material will be dissolving in the total cell. So here we have seen that again the reappearing of the nuclear envelope forms. The nucleolus also starts forming, and we can see that the centrosome organizes itself above the nucleus. Now here we remember the centrioles. In the previous two slides we have seen the green color centrioles were here, from which the spindle fibers were there. So now if we recall the prophase, the opposite thing is happening here. And here what is happening? The centrosome also is formed outside this nuclear cells. The nuclear membrane again reappears around the chromatin network, and the nucleolus appears. And the spindle fibers, here where the spindle fibers, they have disappeared. So this is the last phase of cell division, mitosis. And here see the cytoplasm, they also start dividing into two daughter cells. So division of cytoplasm is called cytokinesis. So it begins during the late anaphase and is completed soon after the telophase is over. So at the end of telophase, what will happen? These two cells will be two different cells. They will be all divided into new daughter cells having the same content as what was there in the parent cell. So actually this process is a bit different in plant and animal cell, but here we are discussing about animal cell. And this last phase where we can see the cleavage is there in telophase, end part of telophase, where it splits into two daughter cells. So this is the last phase of mitosis. So till now I think it is clear for everyone. That is, first we have started with the interphase, then prophase, metaphase, anaphase and telophase. How this nuclear materials were dissolving, formation of spindle fibers, centriole, then moving towards the poles. And then again the formation of the cleavage of the cell, the breaking of the cytoplasm into two different cells has happened. And here we can see the formation of two new different cells at the end of this mitosis cell division. Now, till now, if you are having any queries, any doubts, please do write in my comment box, and please don't forget to subscribe to my channel so that the next updates what you are getting on this lesson also, you can study it and continue with my lessons. As of now, the last slide which is left is quite the importance of mitosis is left, and we will wind up with the brief significance of mitosis. So let us see what is there. Now let us quickly discuss about the significance of mitosis. Now what is mitosis doing? It is maintaining the same number of chromosome of all the chromosomes in all the cells of an individual. Now in other words, we can say that it is an equational division. We have told in the first few slides also, it is an equational division, as the two daughter cells produced are identical. We have seen in the previous slide also, they are identical to each other and even to their parent cell. And this type of cell division usually takes place in the somatic cells, that is the body cells such as the tip of roots and stem, etc. Now next point, we can say that it is playing a significant role in the replacement of cells which are lost during wear and tear and in wound healing. And now it is also responsible for the growth of an organism. And now as a fertilized embryo or a cell develops into an embryo and finally it develops into an adult as a result of this mitotic division. Now it helps the cell in maintaining the proper size, and then we can say that it is a method by which a sexual reproduction in unicellular organism is happening. And when if a mitotic cell division becomes uncontrolled, it may cause tumors or cancerous growth. Now this is all about the main significances of mitosis. Now this is all I have made you understand about the different phases of mitosis and all. And I think you should go through your textbook, whatever book you are following, go through it and try to recall each and every stage with the diagram, whatever I have taught in this video, so that when I move on with the cell cycle, which I thought of teaching in this slide only, but I couldn't continue because as it is a bit long, so I'll be continuing in the next slide, that will be only cell cycle. So there I'll take your comments, I'll take whatever queries you have. So you will put your queries in the comment box, and I'll answer the questions in the next video of cell cycle. So please do subscribe to my channel, and don't forget to like and share so that your friends can also get benefited from this video. Thank you.